Our next speaker is Andrea Luppi, a PhD student in clinical neurosciences, working in Professor David Manone's lab at the University of Cambridge. Andrea's presentation today will highlight the relationship between different regions in the brain during neural information processing.
Hello, hi, and welcome, and thank you for your interest in my work. So I'm Andrea Luppi from the University of Cambridge, and my PhD is about understanding how our species got from here to here. And of course, that is thanks to the human brain. Now, to understand the way that we think about the human brain, it's helpful for us to go together through a little murder mystery. Imagine that you are a detective trying to figure out which of these individuals committed a particular murder. Now, as a detective, you may have found a witness who tells you that the culprit belonged to the suit of hearts. This lets you eliminate some of the potential suspects. And if in addition, your witness also tells you that your culprit was a man, you can identify some further suspects to eliminate. But still, you don't uniquely know the identity of the culprit. However, you may also have a second witness who also tells you that the culprit belonged to the suit of hearts, but adds the information that they wore a crown. Once again, on its own, this information would only let you remove some of the suspects, but not all of them. Crucially, however, if you put these pieces of information together, you can now uniquely identify the culprit as the King of Hearts. Now, the reason why this is important is because you can tell that there are different kinds of information that were being provided by the different witnesses. Some of this information, like the belonging to the suit of hearts, was redundant because each witness provided that information, meaning that you would have had access to it, even if one of them had forgotten about it. Some of the other information was uniquely provided by one or the other witness. But most importantly, the information about the identity of the culprit was only available when the information provided by the two separate witnesses were combined together. That is, there was a synergy between these two witness testimonies. This is important because we now have the mathematical tools to quantify in a formal way synergy and redundancy in information provided by different sources. And this is possible over the last 10 years thanks to the tool called information decomposition. And my work is about trying to identify within the human brain where is synergy most prominent. And in particular, to do this, we used functional MRI data obtained from human participants and made publicly available as part of the Human Connectome project. And this, using the tools from information decomposition that we just went through together, allowed us to identify in the human brain where synergy and redundancy are most prominent. And in particular, as you can see here on the bottom right, the distribution of synergy shown in red and of redundancy shown in blue is not random. And using a meta-analysis of several thousand published studies, what we were able to find is that they also have different associations with different kinds of tasks, such that redundancy is most associated with sensory and motor low-level cognition, whereas synergy is especially associated with high-level cognition. And this association is not just within the human brain, but rather it also holds across different brains. So we compared humans and macaques with data from functional MRI, and what we were able to find is that the two species do not differ in terms of the proportion of total information that is explained by redundancy. On the other hand, we also found that humans are significantly more rich in synergistic information compared to macaques. And the evolutionary relevance of synergy does not stop there, because we were able to find that synergy is especially prominent in regions that have expanded the most over the course of evolution, and it is also especially prominent in regions of the human brain that are especially enriched for uniquely human-accelerated genes. Now, we wanted to go a bit deeper and find what are the underlying neurobiological underpinnings of synergy. And what we found is that synergy is especially prominently associated with synapses, which are the locations, the physical locations that enable neurons to communicate and integrate information with each other, which is what synergy is all about. And we got there through two convergent lines of research. First, we carried out a data-driven analysis of gene expression patterns. And what we were able to find using these post-mortem gene expression data is that regions that are especially synergistic are the same regions that are especially enriched for synapses and synaptic transmission. And we confirmed these results using in vivo synaptic density mapping through positron emission tomography, which showed once again that the more synergistic a region is, the more it expresses synaptic density. So just to summarize our research, what we found is that neural synergy supports higher cognition in the human brain. And within the human brain, it is also especially associated with evolutionary expansion and especially associated with the expression of evolution-related genes. In addition, we find that synergy is especially enriched in the human brain compared to the brain of macaques, which is not the case for the other main kind of information, that is redundancy. And finally, looking at the neurobiological underpinnings, we were able to find that through the divergent and convergent means, synapses are the macroscale and microscale underpinnings of synergy in the brain. And overall, we think that this is prominent and promising in terms of establishing a way to identify what makes the human brain so special. Of course, this was a synergistic effort, and I wish to thank everybody who contributed to this, both people and institutions. And in particular, I wish to express my gratitude for my funders, Gates Cambridge, and also to my supervisor, Dr. Stamatakis from the University of Cambridge. And finally, here is how you can contact me, and I look forward to answering your questions. Thank you for your attention.
Andrea, thank you very much for this deeply synergistic work, as you kind of said. So as always, please feel free to leave questions for anyone who's watching who might want to ask Andrea something into the chat. But again, I guess I'll start it off. So this isn't my field, but I'm very fascinated by this idea that you can look across different organisms to see, actually, this is where some of our higher order functioning as humans can kind of come in, just by looking at different regions in the brain and how they're overlapping. How much is this conserved to some extent as we move further back in the evolutionary tree? Like if you looked at mice or something like this, would you find results like this, a continuation of this trend where there's less density in these overlap regions?
I think that's a great question, and I just want to say it's a real pleasure to have the opportunity to discuss this research with the other attendees. So going to your question, that's something that we're actually interested in delving deeper into. Right now, we were lucky to have the ability to compare humans with a relatively similar species, macaques, who we can scan in a similar kind of environment. Of course, the situation becomes more challenging as the species diverge more, because then it's not clear how much of the differences can be attributed just to differences in information capacity and how much instead depends on other anatomical aspects. But it is exactly the right kind of question that we're also very interested in.
So then I guess I might continue this, and I know it's not trivial to find model organisms, so I'm certainly not saying go out and find these organisms to do it on, but in other kind of kingdoms of life where you have evolution of increased intelligence, if you were to compare, say, fish and dolphins, do you think you would also see a similar increase in that particular, those particular characteristics you saw on the primate chain?
I hadn't actually thought about the comparison with marine life. I think once again, the differences between fish and dolphins might not be just in terms of brain size and cognitive capacities, because of course, dolphins are also mammals. So there is also that additional part of the differences that might need to come into play. But we do think that there is something that has to do with, in general, what synergy enables any information processing system to do. And to the extent that we can refine that from a theoretical standpoint, then I think we will be in a better experimental situation to be able to ask exactly what kind of question we can predict that would be related to synergy and obtain an understanding that is independent of the implementation, if you will, and just really depends on the capacity of the system itself.
Sure. So the synergy really might be sort of universal as we talk about an increase in intelligence. That's fascinating. So Nishema Gilad says, great talk. What are the plasticity rates of synergy? Are there any changes with age, for example?
That's also something that we are looking into right now. So the data that we have come from the Human Connectome project, which is a data set of relatively young and healthy adults. But for instance, we have some follow-up work that is looking at what happens in patients who have suffered a disorder of consciousness. So they've, for instance, in a vegetative state as a result of brain injury. And in that cases, what we see is that there are changes that involve synergy as well. So that is not the same as answering the question of what happens with aging, but I have colleagues, some of my co-authors on this paper, in fact, who are also looking at gradual changes in a very large database called CAMCAN that comprises ages from 18 to 80. And there are changes that occur both in terms of synergy and in terms of redundancy. So part of the question now is, how do we interpret them? What do they mean? But there do seem to be changes of this kind, yes.
Okay, great. We also have a question from Anonymous, who says, do we know what could interfere with or improve such synergistic neural interactions?
So something that we also know, in addition to brain damage, which of course happens to have a lot of different changes on brain function, but we have also compared that with the brain changes that happen when someone undergoes anesthesia, which is something that happens millions of times every year in the world for, of course, surgery. And luckily, it is reversible. People are anesthetized and something changes in their brain, but then as the drug wanes, its effects also wane and people come back. And we have seen that these effects are relatively similar to what someone who is in a vegetative state will have. But the difference being that in the case of anesthesia, this is a transient change and then everything goes back to normal. So that tells us that synergy can be perturbed pharmacologically, but that this perturbation will also be reversed once the pharmacological intervention is wanes off.
So I kind of want to follow up a little bit on this. Do you think it's possible for us to train the synergy in our brains? If you tracked a single person, as they do the crossword puzzle every single day for 2 months or something, would you see an increase in that density, more synergy in the brain? Or do you think that each one of us have our own kind of levels?
That's a fascinating hypothesis. I don't have evidence right now to say it's for or for against it. But I do think it would be interesting because, of course, you mentioned what happens with aging, but something that we're also very interested in is how synergy develops into adulthood and throughout, from fetus to newborn to then into adulthood. Does that change with education? And that's something that we currently don't know, but I think it would be very interesting to find the answer to. And likewise for artificial systems. We currently don't know whether different ways of training an artificial system might be especially suitable to induce synergy there with its advantages and all of that. Yeah.
Excellent. So we have a lot to look forward to. So thank you very much, Andrea. And now I think we'll move on to our final talk of this session.
Thanks.